everybody. So today we are going to work on part 2 of diffusion through a membrane state lab. Part 2 discusses and models for you the action of osmosis, which, as you know, is the diffusion of water through a membrane. So the materials that we will need today include your tweezers, a cover slip, a slide, a red onion, fresh water, and salt water. So we're going to start by placing some fresh water onto the slide. And we'll also need some paper towel. And then you are going to receive onion slice like this. And in order to get this dark membrane here onto the slide so that we can observe it under the microscope, we're going to actually split it in half like this, and you'll feel that crunch happen. And then when you split it in half, you're going to gently tear the purple part of the skin so that you can see those cells clearly under the microscope, just like this. So you want to get in the fleshier part and then gently just peel away, and you'll notice the skin coming off. And you're going to gently just peel away again until you get a nice sample of that onion. Wow, this is really big. Okay, so then you're going to gently place it using the forceps, and you want to make sure it's flattened onto your slide. You don't want it to double over, so just be careful with that. Gently place it onto the slide. And then you're going to take your cover slip, and as you know, we hold the cover slip at a 45-degree angle so that we avoid air bubbles under the cover slip, so it's easier to visualize. So the best way to do that is to use your pencil and gently hold up that cover slip and then gently place it down, just like that. And I'm just going to use a little bit of water to clean up the side. Just pull it up. Notice how it quickly just gets absorbed, but you don't want to get too much water because you still want some of the water under there so that you can see a clear sample. And so this covers steps 1 through 3 in the lab. You are now going to complete steps 6 through 7, and you're going to draw a sample of one of the cells that you see. You can draw more than one sample, but just make sure that you label one of the samples. And since the onion cell is a plant cell, you want to make sure you label the cell wall, the cell membrane, and you're going to see dark spots. Those dark spots somewhere in the cell, that represents the nucleus. So you're going to make sure you label those parts. Once you've viewed it under the microscope, you want to call me over so that I make sure that you're actually viewing the onion cell. And then you're going to go ahead and draw your sample in the packet. And then we're going to continue on to the rest of the steps. Make sure you follow directions in procedure number 6. Based on the observation, draw and color a typical red onion cell mounted in water. Make sure to label the cell wall, the cell membrane, and the cytoplasm. As you see here, this is something that you should be seeing under the microscope. Also, make sure if you do see the nucleus to label that as well. So now we're going to use a similar technique that we used for staining the onion cell, except here we're adding salt water. So we're going to take our container labeled salt water, and we're going to place on one side of the cell about 5 drops of salt water. And then I'm going to put the paper towel on the other side, and I'm going to allow that to just come across the cell. I don't want to take up too much of the salt water because I want to be able to observe what happens to the cell when I have added the salt water. I'm just going to clean it up on this side a little bit. Okay, just one more time to make sure it has enough salt water. Let me get another piece of paper towel. We're going to add the salt water on one side and then absorb it on the opposite side, right? Notice how the water is moving across onto the paper towel. And then you're gonna go ahead and draw those observations that you see under the microscope and pay attention to what happens to the cell membrane and how it moves away from the cell wall under the microscope, and make sure to label the cell wall, the cell membrane, and the cytoplasm. Then you're going to answer questions 11 through 13, and then we're gonna add fresh water again. So now you will turn to question 10 on page 7 and sketch a picture of the red onion cell in salt water. Make sure to label the cell wall, the cell membrane, and the cytoplasm. Take notice of how the cell membrane has moved or pulled away from the cell wall. Make sure to explain why this is happening in question 11. Don't forget to label the cell membrane, the cell wall, and the cytoplasm. Again. So now you're going to be able to see the change in the cell when you add a hypotonic solution, a hypertonic solution, and then a hypotonic solution again. So this water is pure water. There are no other molecules in it, and that's why it would be considered a hypotonic solution. So like the previous procedure, we're going to add that water on this side, and then I'm going to have that pull away with the paper towel. And I'm going to just do this one more time to make sure that the solution has gone across properly. Once you've completed this, please also make sure that I view both the salt water and the distilled water under the microscope to make sure that you're doing this properly. Draw your observations, label the cell wall, the cell membrane, and the nucleus, and then you're going to continue answering the analysis questions in the lab, 1 through 6. Okay, have fun.